Professor at Department of Industrial Chemistry, Aligarh Muslim University. My dear students, Assalamu alaikum and a very good morning. Today, on the last day of this 5-day GAN program, I welcome you all. And in the morning session, we'll be having a very nice and meaningful and very interactive type of session with Professor Liu. So I urge all my students to keep an eagle eye and to get involved. So with this, I invite Professor Hitao for the lecture. Thank you.
Thank you for the kind introduction. Okay, this should make the Zoom to work now. Okay, all right. Thank you for coming back. I understand today's a holiday, so I appreciate students and faculty giving up the precious personal time attending this sequence of lectures. Today I'm gonna, well, in this lecture, I'm gonna provide overview and introduction of DNA nanomaterial. This is a new emerging and rapidly developing area of research. So my talk in the morning would be more on the general overview, and we'll spend more time on the manufacturing, fabrication of those nanostructures, the chemistry principles behind those construction of those structures. I will briefly touch on the applications, focusing on the unique properties of this class of materials. In the afternoon, I will, well, the lecture after this one, I will focus more on my own research program in this area, in particular using DNA for large scale and high resolution nano fabrication. And in the afternoon, I will be able to show you some videos recorded in my own lab, showing how students in my lab are making those DNA structures, but also characterizing them using atomic force microscope. Uh, we have some technical challenge. Maybe just show the
PPT first or video?
just show the PowerPoint. When I show the video, I'll just switch back. Good. All right. Hopefully, you can colleagues online can see the shared screen now. Okay. So if you missed my previous lectures, this is where I come from. I am a faculty member at the University of Pittsburgh Department of Chemistry. This is located in the eastern part of the United States. Pittsburgh is it's the second largest city in the state of Pennsylvania. Let's see, pointer options. And it's located in the western part of the state. So before I jump into the topic, I want to do a little bit of flashback and give you a little bit of context here compared to the nanomaterials that I talk about in the previous days, but also you probably are very familiar with. And I also was asked a number of questions about different forms of nanomaterial. So I want to briefly talk about why DNA and how DNA nanomaterial is different from many other nanomaterials. So quantum dots or nanoparticles, either metallic, if it's metallic or oxide, usually people just call them nanoparticles. If it's semiconducting, often semiconductor nanoparticles are often called quantum dots. And the reason being, those are small small particles with their size around 2 to 100 nanometer. Now, why semiconductor nanoparticles are also called quantum dots? Because there's a phenomena called quantum confinement. You have a wave function has finite size. In a bulk material, the size of the material would be greater than the size of a lot of the wave functions you are dealing with. In that case, the material property is not limited by the material itself. Okay? Now, when you have a much smaller particle, thinking about 2 nanometer particle, but the wave function in some cases can expand 2, 3, or even 4 nanometers. Now you come to this interesting geometry and size regime where the size of the material would impose a limitation property. And you can actually change material property by simply change the size. In another way of seeing this is that there's a quantum confinement in the nanoparticle if the size is right. And that quantum confinement is happening in all the 3 dimensions, x, y, and z. Another way of looking at this is a 0-dimension material, meaning that all 3 dimensions you have a quantum confinement, and there's no dimension that is not quantum confined. So that is where the 0 dimension comes from. We also talked about carbon nanotubes a couple of days back. Now those are tubular structure. If you think about material in terms of geometry, you can view this as quantum confinement in 2 dimensions. That is x and y, but not z. If you think about carbon nanotube along in the z direction, so wave function can freely propagate in the z direction, but confined in x, y. And again, this will produce a modification of electronic structure and band structure compared to the bulk electronic structure. So in the case of quantum dots, light emission has been a very popular application. You may have seen commercials of Samsung TV, LG TV. Sometimes they label their product as quantum Q dot TV, or terminology like this. And what is happening is they actually put cadmium selenide quantum dots in their display, because those quantum dots can produce very pure color, very pure red, very pure green, and very pure blue. So those 3 colors will reproduce all the possible colors out there. In the case of carbon nanotube, there's a similar property in terms of modulation of emission, but most of the applications are in electronics, where semiconducting properties are being taken advantage of. Being one-dimensional also creates additional benefits in terms of making device, because a lot of devices, well, pretty much all the devices have finite size. So when you're dealing with small particles, you have to think about junctions between particles. If you're dealing with a tube, then there's no such junction you have to think about. So that makes it easier and more suitable for manufacturing nanoelectronic devices. Now it goes to graphene 2D materials. Those are materials having very narrow thickness, but in the x and y direction, they can be infinitely large. Okay? So those are materials having quantum confinement only in 1 dimension. So instead, they're called 2D materials. 3 minus 2 is 1. Okay? So 1 dimension of quantum confinement, and the other 2 dimensions are not quantum confined, and they're called 2D. In the carbon nanotube, is 1D, meaning that the other 2 dimensions are quantum confined. In the case of quantum dot, all 3 dimensions are quantum confined, leaving them as leaving them a name of zero-dimension material. Now, those materials are all very interesting in the sense that material properties can be a function of size. Okay. The DNA material I'm going to talk about today is a little bit different. In this case, the basic building block of the material is double-stranded DNA. This is the foundation of life. If you think about the double helical structure, it's about 2 nanometer in diameter. Okay. You can view it as a wire, essentially. What I'm going to show you later is how you can start from this very simple wire structure to construct higher order structures as large as several hundred nanometer in dimension. Now, this class material is covalent in nature. It's covalent, but also there's certainly covalent linkage within the DNA chain, but also there is non-covalent interaction, hydrogen bonding electrostatic that pull the chain together. There's no quantum confinement in this case because DNA is pretty much an insulator by most people's standard. So we're not looking at excited state dynamics. We're not looking at many things that we typically look for in a quantum dot carbon nanotube or 2D semiconducting 2D material regime. There's no light emission, at least not very strong light emission. Instead, we're looking at many other properties that are fascinating, in particular, the exquisite control over the size and the ability to manipulate the location of different material components. So if I have to summarize this slide, there's a big difference in terms of motivation, why we want to study those material, and what are the unique strengths of each kind of material. Okay, so all this business happened about 40 years ago. The person on the left, his name is Nat Seaman. He was a professor at New York University, chemistry department. And he's considered by almost everybody in the field to be the father of DNA nanotechnology. He actually has a PhD degree from University of Pittsburgh. So he's actually our chemistry department outstanding alumni awardee. And he was trained as an X-ray crystallographer. So if you recall the history of United States, 50, 100 years ago, there's heavy industry, steel manufacturing, heavy metal processing. So Pennsylvania, being an industrial harbor at that time, has a department of crystallography. Okay, so students studying how to do single X-ray, structure, refinement, things like this. So he got a degree in doing X-ray diffraction. Now that department later merged into chemistry department, which makes us consider him being our alumni. Now he's actually he's not a DNA person, but rather his interest is to think about resolving protein structure, and to do that you have to grow protein single crystal. And he started his independent career in NYU, New York University, around late 1970 or early 1980s. And he spent his time resolving different kinds of protein structure, basically. But it's frustrating. I don't know if you have done those, but proteins are big molecules. It's difficult to crystallize them, very difficult. So he would be frustrated not being able to get protein crystal, can't figure out how to do them. If he doesn't have a crystal, then he cannot do X-ray study. So, and one day, this is a story told by him, and one day he went to a bar near the university campus. So he is thinking about a research problem, how to make a crystal of protein, because X-ray happens only when you have regular array of materials, right? You have to have this lattice. But growing protein structure into a single crystal is difficult. Now, how can he get around this problem? It turns out, well, just as fate may have it, in the bar there's this art. the one hanging on the right, I think it's called something like the depth or something like this. I'm not an artist, so that name of the picture escaped me. But it's a pretty well-known art piece produced in the 1950s. And it shows this array of fish. Okay? And the array extends in 3 dimensions. And each fish has this fin in in 2 dimensions, and they're somehow aligned together. Okay, so to the eyes of an X-ray person, this is a crystal. You have 3-dimensional repetition, and suddenly somehow lightning strikes him. Whatever happened, and he started to think, "Oh gosh, how can I make artificial lattice, a 3-dimensional lattice? If I can do that, can I just attach a protein to each of the units? And if I can do that, then I will have a way to position protein in a regular lattice structure. Then I would have a crystal. I would have a crystal not dependent on the particular shape of the protein, but rather dependent on extra sets of framework. So just thinking about all the scaffold you encounter in a building a high-rise building, you can always make this large terrace structure, and if you can put molecules at exactly the same location within each unit cell, then you can create an artificial crystal. So this became the starting point of a wave of evolution in structural DNA nanotechnology. And this very simple idea of using something to create a molecular lattice framework become the underlying principle and thought and philosophy of DNA nanotechnology. So after that, he just started thinking about how he can use double-strand DNA to create this lattice structure, and how he can then start from there, attach different molecules onto this DNA lattice, and then from there, a lot of other things happened. Turned the research into something he never imagined, a very productive research area. Unfortunately, he passed away 4 years ago. Before he could have won Nobel Prize, in my view, if he lived another 20, 30 years, probably he will won another Nobel Prize in chemistry. All right. So looking at the very fundamental structures, now this is a little bit more biochemistry. What I've shown here on the bottom is the bottom right is the conceptual idea that Nassim had in the 1984, I believe. So here, I show this lattice structure XYZ, and here's this some kind of blob, greenish, bluish blob. This is the sort of the protein that I want to attach to the lattice. If you can attach it to the same space, same area, same location, then you can come up with some kind of cubic lattice of protein. Okay. Now, how can you do this? Well, what Ned Simon thought about very early on was some kind of junction, a DNA junction, and one type of junction he worked with in particular was so-called holiday junction. And what this is essentially four strands of DNA come together. And you look at the structure here. This is the schematics. You have 4 DNA strands, and each DNA strand can be broken into 2 segments. And each segment is recognized by another segment of another DNA strand. So if you have a structure like this, you can essentially combine, I'll reject. You can combine 2 double helical structures together. So essentially, you are threading 2 wires into one. Okay, that's the way I look at it. Now, if you have, and the way you can draw them is just like a cross. Okay. But another way of looking at it is you have double helical structure on one side, another double helical structure on the other, and in the middle you have two crossover. Okay, the two in the middle is a crossover structure, and this crossover structure brings the hydrogen bonding structure together. So instead of have one wire, you have two mechanically interlocked together. Now you can design this structure to have multiple crossover. You can also design structure to have so-called sticky ends. A sticky end is elongation of, is shown here. So if you look at this motif, one end of the, one part of the double helical structure is elongated a little bit longer. And this produces a little bit of additional single strand motif. Now this one will be free to hydrogen bond to other parts of single strand DNA. Okay. Now imagine if you have a lot of those things, you can create, those are essentially cross structures, but can polymerize onto themselves. As long as you design the end of the sticky ends in a particular way to recognize themselves, then you can polymerize this in 2 dimension or in 3 dimension, so on and so forth. And once you can do that, you would have a 3-dimensional, 2-dimensional DNA lattice. Okay. So this is a very rough idea that Nate Simon put forward in the mid-1980s. Once people got into this, concept, things become things start to evolve rapidly. So here, what I show a so-called tile-based structure. Why I call it tile? If you look at this this crossover, and this crossover is another upgrade of what I show here. Here you have one crossover, right? But you can have multiple crossover within the same sequence, well, within the same hybridization, I guess. So here, for example, you have you can view it as two double helix DNA in parallel, but it has one crossover here and another one here. There's actually there's a circular one here as well, and this is called DX. You have two become one, and you can do it over even larger lateral dimension. Imagine you are making a carpet from threads. You put a threads in parallel and somehow connect them together. This is exactly what you're doing, except you're doing it using molecular scale wire and a thread. So once you do this, you can make a larger structure, and you can play with different kind of crossover. Instead of having an individual, you could have two together, something like this, and this can create improve the mechanical property of the nanostructure. So using this concept, you can using them as a building block to create much more complex structures. And I show some examples here. This is a small building block produced by multiple crossovers. And also, at the end of the building block, again, you see this small niche, small hangover, just 2 bases long. Okay. Now, the sequence here is designed in such a way that they will find themselves and polymerize themselves in 3 dimensions, X, Y, and Z. Once you have a lot of the structure in solution, and this is an X-ray structure of the unit, they can, as I said, they can polymerize in X, Y, and Z and actually produce crystals. Okay. And shown here is an optical photograph of the DNA crystal, a porous DNA crystal being produced using this technique. This is exactly what Nassim envisioned in 1984 or 85. Took the community about 30 years to make it work. You can also, using this technique, instead of folding it, instead of polymerizing onto themselves, in that case you get a lattice, X, Y, Z dimension lattice. You can also have the structure to recognize another portion of itself. So if you do that, here's one example. You can use this fundamental building block to create intermediate scaffold, and in the intermediate scaffold would recognize either themselves or with another scaffold, and by doing that you can create polyhedral structures, and those would be on the order of several nanometers or several tens of nanometers. Okay. And people have, tried different versions of this, but the essence of this slide is that you can use this very simple tile or tile monomer to create individual DNA structure or lattice of DNA structure. Now, the research took a very, I would say, unexpected turn, but in the positive direction. In around 2006, and a group, one scientist at Caltech, California Institute of Technology, published a paper in Nature with only one author. That person is the sole author of a paper in Nature. Incredible idea and demonstration of a concept called DNA origami. Now, what is this origami? This is a very different way of making and designing DNA structure of pretty much arbitrary shape. So what I showed in this slide is some kind of repetition lattice. That's interesting, but what if you want to write your name using DNA? That's much difficult to design here, right? Because here you have a lot of constraint that you have to work with, and there's no way of scaling this up easily. This scientist at Caltech, his name is Paul Rothman. He developed a very different idea, and his concept is to taking the hint from origami, which is the folding of paper, and he took it into the nanoscale world and thinking about folding of the DNA strand. So on the top left is the concept of this DNA origami approach. And what I showed there is a lot of wires. There's a black line that goes through. So there's a black line, and you can imagine this is a very long single-strand DNA. Okay. And there are bacteriophage DNA that are several thousand bases long, and this is one of those that you can use. And this strand is called, in the community, called scaffold strand, because it is actually being used as a scaffold of the construction of a DNA structure. What is also present are a lot of colored lines. Now, those color lines, those are also DNA strands. They're much shorter in sequence. Usually, they're about 20 to 40 bases long. But there are a lot of them. And I also want to emphasize that each one of them is unique in sequence. Okay. Now, what is happening here is that you have a very long DNA strand, and you know the sequence, you know the whole sequence of that long DNA strand. Okay. Think about 7,000 bases long. You can create a lot of staple strands, those short strands, each being about 30 to 40 bases long, but you make a lot of them. In reality, you make about 300 of them. So 300 DNA strands, each one being unique. What you can do is you can design the sequence of a staple strand such that two portions of the staple strand would recognize two portions of the scaffold strand. But those two portions on the scaffold strand can be really far apart. Okay. Now imagine what's going to happen when you mix the two together. The DNA would perform hydrogen bonding. As I said, the two portions of a short strand would recognize another two portions of the long strand. But because the two portions of the long strand are so far away, what's happening is the short strand essentially burn the two parts of the long strand together. Now, essentially what is happening is you are folding the long DNA strand by using a short DNA strand, the staple strand. So you're stapling the long DNA by folding them in half. What is powerful in this concept is because there are a lot of short strands, as I said, about 300, you can apply this concept in an iterative fashion. Essentially, you can fold the long strand 300 times. And this is exactly what is happening here. Okay. By folding it 300 times, you can start with a very long wire, seemingly having no structure, but you can fold it in any way you want. You can fold it any way you want and fold it 300 times. Imagine if you have a piece of paper, you fold it 300 times, you can produce any shape you want. Same thing happens when you fold a piece of wire, except in this case, you got a two-dimensional shape instead of a three-dimensional shape when you do actual paper origami. So if you look at the pictures here, okay, the pictures here shown are models of DNA structure, and on the bottom are actual structures produced by this technique. Each one of the structure is about 100 nanometer in size. Okay. To give you a context, your hair is a couple of micrometer, so this is about 1,000 to 10,000 times smaller than the diameter of your hair. Now, even with that kind of small dimension, you see very exquisite control over the shape, and this smiley face being one prime example. You can essentially fold it into any shape you want within that limit of 100 nanometer by 100 nanometer size regime. Using the same kind of approach, you can also create a tile. So the B panel shows a very similar origami approach, except it's a little bit connected, a bit more connected to the tile-based structure. Here, what they did is they create this regular tile, and then they fold the tile into defined pattern. And this is more like a piece of paper being folded back and forth, back and forth. But the same kind of idea. You use short strands to recognize two portions of another structure and fold them into geometric shapes. If you look at the bottom, you see that this kind of concept can also be applied to make three-dimensional structure. On the bottom, there's some kind of ring structure that mimics a gear, a mechanical gear, except it's probably the smallest gear you can make with a size of around 100 nanometers. On the right-hand side, it's even more interesting. This is a nanoscale flask. You can fold everything into an irregular shape in the 3-dimension. And this whole thing is about 40 nanometer in this dimension. So a very exquisite control of the folding of very flexible material. And that is, in my view, the true evolution of DNA nanotechnology. It really enables people to make arbitrary shaped patterns on both 2D and 3D. To summarize this slide, I want to recap the two main components. One is a scaffold strand. This again is a very long single strand DNA. And one of the most often used one is called MP13. That is a bacteriophage DNA, about 7,000 bases long. And then what you need is a lot of short staple strands, depending on the size of a nanostructure. The typical structure made here requires about 300 unique DNA strands. Okay. Again, each one is unique. Okay, I'll pause here for a bit and to see if people have any questions on the construction. Are you talking about the fold sequence? Okay, that's a very good question. So if you think about this kinetic process here, you have one long strand DNA and 300 different strands. In reality, what happened, when we make them, in the afternoon I'll show you a video of this, we actually mix all 300 strands and the scaffold strand together. And we don't even care too much about the ratio of them. And we heat it up and cool it down, and magically the structure is being produced. So if you think about this being a stepwise process, it feels almost impossible because there are so many different ways this could go wrong. And indeed, there is a lot of different ways this could go wrong. But at the same time, this is probably more a collective and self-catalyzing in some way. Self-catalyzing is probably the wrong word. But there are many different pathways, and it doesn't have to be folded by a particular sequence, but rather any one of the 300 strands can fold the long strand, and they just go through different pathways, but the final structure energetically is so favored, so it always go back to that location. And also, this would require very slow annealing. So we typically anneal the reaction mixture for 10 hours or 20 hours, very slow cooling. So, so the structure has a lot of opportunity to fix the problem. There's dynamic equilibrium between hybridization and non-hybridization. If the structure got stuck, it can always go back and find another pathway to eventually go to the end. So it doesn't happen in every case that you will produce high yield, but Rothman's original approach is very easy to reproduce. My lab has no experience of DNA nanotechnology. When we started doing this, we reproduced their work in our second try. Any other questions on this? Okay, you can always ask the question at the end. All right, so the origami approach has been very popular and very powerful, but people also started to think about different ways of making the structure. And if you think about the origami approach, one of the biggest limitation is actually the scaffold, because you need this very long single-strand DNA, and the length of that DNA actually limits the size of the structure you can make. Okay. But also, in addition to that, because the folding is, if you fold a square, you can just do this, right? If you fold a triangle, you have to do this. Okay. So the folding sequence would be very different depending on what kind of shape you make. So to make each one of those shapes, you have to make a separate set of staple strands. So what that means is if you want to make smiley face, you need to go to the DNA synthesizer company and buy 300 strands. Okay. If another day you want to make a triangle, you have to buy another 300 sets, and those 300 sets are totally different. Okay. Now, fortunately, there's a computer program to design everything, so you don't have to, work on paper and pencil to figure out what sequence you order. You can just put your shape into the program. Program will tell you what sequence to order. You can just order them. But that still is very inefficient, both from a material utilization, but also a compatibility across different shapes. So about 10 years ago, there's another approach being reported. This is called single-stranded tile approach. And it's also being referred to as DNA break. It all depends on the context you're using this approach. Now, this approach has the most significant feature of not using a scaffold. So in this case, everything, all the structures are produced by using a mixture of short DNA strands. Okay. And each DNA strand is the same length, 32 base pairs long. And this is one example. Okay. Imagine what you have here is a 32 base long single-strand DNA. You can artificially break it into 4 domains, okay. And each domain will be 8 bases long. Okay. What you can do is you can make, let's say, 10,000 of those strands. Again, each strand would be different in its sequence. But what you can, in the design phase, you can make the sequence only recognize its neighbor. If you look at this plot shows a bunch of single-strand DNA. Again, each one has 4 domains. Okay. If each one has 4 domains, you can think about in the XY dimension, each DNA sequence can recognize 4 nearest neighbors by using each of the 4 domains. Okay. If you can do that, then you, by designing the sequence, you can have DNA sequence 1 and 2 sit next to each other in this direction, and sitting next to each other in the vertical direction with different neighbors. Okay, so that's the idea here. You have 2.1, 2.2, 2.3, and it's designed in a way that that each one of them knows where they should be, because only if they are at the right place, their bases can hydrogen bond with all their neighbors. If they're in the wrong place, then they cannot hydrogen bond with all their neighbors. So in a sense, it's like 1,000 students come to the come to a big room, and you don't tell them where to sit. Okay, let's say we have 100 people here. Let's say, and I'm not going to tell you where to sit, but I put everybody into a room. I only tell you you need to sit next to your best friend A and B in this row. On your left should be A, on your right should be B. And in your front should be C, and in your back should be D. I'm going to tell everybody that information, and I'm just going to let you to figure out. And eventually, you will figure out how you should sit together. And the information I tell you would be the sequence information of the 4 domains. So by encoding that information in a specific way, I can let the DNA sequence know where they should be next to which sequence. Okay, so that's the idea. Now imagine if you have, let's say, 100 of those sequences, you can make them into 10 by 10 array. And that would be just a canvas, a piece of paper. You can make a flat, square-shaped nanostructure. And that would be something like this. Okay. Again, in this structure, each of the component knows where to find itself within this bigger structure. Now, what is more interesting now is if you eliminate some of those strands. Okay, if you eliminate some of those strands, for example, if I look at this structure and I'm going to draw a line and I say I'm going to get rid of all the strands on the top left, but I still keep all the strands on the bottom right in the annealing process. So in that case, the annealing will still happen, except it will only happen to produce a triangle shape. Okay. You can also say, I'm going to get rid of the strand that's supposed to be in the middle. And you do the annealing without that strand, you're still going to produce this rectangle structure, except in the middle would be a hole. Okay. Now, if you are being strategic, you can create shapes like all the alphabets, all the numbers, and all you need to do is to simply eliminate some of the strands in this tile. Okay. And by doing that, you can create 2D geometric shapes of anything. That is, the only limitation would be the size of the molecular canvas. Okay. And what's beautiful in this construction is that you can use the same batch of DNA sequence to produce any shape you want within that size limitation. You no longer have to design another 300 sequence every time you want a new shape. So all the shapes here, for example, are produced by the same batch of DNA sequence. The only difference is which one you are eliminating and which one you are kept in the annealing sequence. Now, this idea can be extended to 3-dimensional, and the only change you need to do is you need to create another dimension. So here, the 4 domains would recognize each other, but only within the same XY plane. What you can do is to play a little bit trick to make the DNA sequence turn 90 degrees after the hybridization. So once you do that, you will be able to create three-dimensional shape. And this is the same kind of idea. A lot of short sequences, they know exactly where they need to be in a three-dimensional assembly. If you remove some of them, you can create geometric shape. If you include all of them, you're going to get a cube. Okay. So on the bottom are the actual shapes produced by using this DNA break approach. And those are transmission electron microscopy images. On the top are the models, cartoon models, and you see that the actual device mimics the model in a with very high definition. All right, this this approach can be extended into even larger scale. If you design sequence in a way that it can polymerize onto itself, you can also create large-scale lattice structure. And here I show a couple of examples. This one where you have a cross lattice, this is still more or less 2D in nature. Okay. But this one, for example, you have a very large plate. Okay. And on this plate, you have a lot of holes. And this kind of structure turns out to be very useful in making high-density patterns for nanoelectronics application. Okay. All right. So the structures I talked about so far, the DNA origami, their size limitation is about 100 by 100. And the single-stranded tile and DNA break also has similar limitation because if you think about this, each domain here has 8 bases. Okay. Now, how many unique sequences can you get by using only 8 bases? Not many, because there's AT and GC. There are only 4 DNA strands. Right? If you do the math, you end up with thousands, maybe tens of thousands, somewhere there. My math is not very good, but we're talking about 2 to the 8th power or something to that order. So the number of sequences you can play with here is not that much. And once you start to have duplicated DNA strands, then the selectivity becomes a problem. So there are 2 approaches to solve this challenge. One is by doing stepwise assembly. This work was published by a group, again, at Caltech, where they produced, you may recognize this is Mona Lisa, except it's at the molecular scale. And what they did is they start with a small origami structure, and they designed a system where each smaller tile can recognize its nearest neighbor. So it's a combination of the origami and the single-stranded tile approach, except each individual building block become a DNA nanostructure itself. Okay. They were able to produce, by stepwise annealing of those components. They start with 2 by 2, and then 4 by 4, and then 8 by 8. So progressively building larger and larger structures. And this is an atomic force microscope image showing the pattern of Mona Lisa on the surface. So there's a video this group of authors produced. And let me see if I can show this video. I also see a chat. Can you see my video online? Good. Okay. Should I? supposedly there is should be audio. Okay, I can hear a little.
Repeated at different scales, taking
Start.
nature is remarkably good at assembling complex structures such as human bodies or trees using simple building blocks such as cells. One of the principles that allow complex structural formation is called fractals, for example, as seen in this jacaranda leaf. A jacaranda leaf has nearly the same branching rules repeated at different scales. Taking inspiration from nature, we developed a strategy for creating complex DNA nanostructures by applying the same assembly rules repeated at different scales. We started with a square DNA origami tile. We designed how the tiles should interact with each other in a hierarchical, multi-stage self-assembly process. Molecules, in our case, double helical DNA, can be selectively attached to specific locations on the surface of each square DNA origami tile, creating a raised pattern that can be seen by atomic force microscopy. When all tiles are assembled together, from 64 test tubes to 16, and then to 4, and finally to 1 test tube, any complex pattern can be created, such as the Mona Lisa. The fractal assembly approach allows the same DNA strands to be reused for interactions between tiles at different stages of the assembly process. Thus, structures with increasing sizes can be created with the same small set of DNA strands. We developed an online software tool that converts the user's desired image to DNA strands and wet lab protocols. The structures that we created are 64 times larger than the original smiley face DNA origami and are similar to the size of a small bacterium.
Okay, so that this slide shows all the structures created by this group of authors, and as you can see, this is a excellent degree of control, over the molecular scale morphology and placement of materials over about 800 nanometer laterally. So this is a very impressive engineering feat. Okay, so I want to go back to my slides. Okay. So in addition to let me hide this. So another approach is simply have more strands. If you recall the two approaches, the origami approach always required this scaffold strand. Now, if you go to the DNA break approach, that restriction is no longer there. And the restriction instead becomes the size of each domain. So if you make each domain longer, so the original size is 32 base pair. If you make it 52, then that becomes 13 bases for each domain. And with that, you can create as many as 10,000 unique single-strand DNA sequence. And what that means is you can put those 10,000 bases into a annealing solution and have them create larger and larger structures. And this is what is shown here. This group of scientists at Harvard, they created super large DNA brick structure, the largest being the cube, but if you intentionally eliminate some of them, you can create any 3D shape of your choice. So this shows the actual structure. Here the scale bar, I think, is 100 nanometer. So what you see here is a cube structure, not a 2D, but 3D. So it's 100 by 100 by 100 nanometer cube. There's this enormous amount of material packed into this nanoscale material, but also at the same time, within each individual component can be precisely controlled to the single strand level. You can add or remove one of the strands, and if you do that, you can create cavity, you can create three-dimensional shape, and those are the structures people have made, either as a positive tone or negative tone three-dimensional shape. It's like a sculpture using molecular additive components instead of microscopic material. Okay, so that's the discussion on the fabrication. I have about 15 minutes. I want to quickly go through the applications. The application, as I said, is different from the inorganic nanomaterials I focused on. Here, the unique feature is the ability to control the shape. Control the shape and control the positioning of components onto those nanostructure. A lot of the applications are involve some kind of patterning, and I'll spend a bit more time discussing the work in my lab that evolves around patterning, but also assembly of other materials. The assembly can be a molecular species, can be a nanomaterial assembly, can be biomolecule assembly, can be carbon nanotube, can be a lot of things. There's also a lot of research done in medical-related application. I'm not expert in this area, but I'll try to mention some of the interesting examples I saw in the literature. Okay, so on the assembly, there's this is probably the earliest application of DNA nanotechnology. A lot of the early work are actually not using a DNA nanostructure per se, but simply rely on strands. So those are some of the examples in the literature you find in the early days of combining DNA with nanomaterial. And usually you simply modify a nanoparticle. This can be gold or quantum dot, combination of them. And if the DNA sequence are complementary, then you can essentially make the nanoparticle being able to recognize each other or recognizing a different kind of nanoparticle. So what you're doing essentially is you encode personality into a nanomaterial that otherwise does not have any unique name or feature or whatever that might be. With the mediation of the DNA strand, the nanoparticle can assemble into large-scale crystal in some cases, but certainly even at nanoscale, you can create structures like, large and small come together, things like this. With the DNA nanostructure in place, you have even more powerful controls you can apply. Again, because in every DNA nanostructure, you have the ability to control each individual DNA strands. In the origami approach, for example, there are 300 unique strands. You can modify any one of them. You can modify any one of them to recognize any other nanomaterial or any other molecule. So, for example, in this work, the authors created this DNA nanorod. And on this DNA nanorod, because each location has unique specific DNA strands, you can attach gold nanoparticle in a special way, and in this case, in a spiral. And this turns out to be quite useful because plasmonic resonance of gold nanoparticle can be coupled together. And in this case, because the arrangement is chiral, so the coupling is also chiral. So if you have learned organic chemistry, you know chiral molecules often have unique properties, especially optical properties, because you can differentiate left-handed right and right-handed right. Now you can do the same thing with nanomaterial, except the interaction with light is much, much more stronger. There are different versions of this. For example, here you have 2 gold nanorod being templated onto 2 DNA rods, and they can be rotated, and depending on the angle of the rotation, you can get different optical response. And even more examples down here. And the common theme in all the studies is the control, either dynamical. So here's a case of dynamical control. What you can do is you can add or remove some strands, and by doing that, you can change the geometry of the DNA structure in real time. So by doing that, you can alter the coupling between the interaction between DNA nanorods, and from there, you will get a different optical response. Okay. There are other forms of this. I want to highlight this one. This is a case where different size quantum dots are being anchored onto a triangle-shaped DNA structure. And each quantum dot has DNA strand modified. And you may notice the cartoon here, on the triangle DNA structure, each location has been modified with additional DNA anchor strands. So once you put everything together, you will see this, this is the SEM image. You will see the small dots, those are the quantum dots, and they will align in exactly the way you intended. And this is exciting in a lot of applications because now it offers a way to position different nanomaterial in a very specific way. So you can control energy transfer, you can control charge separation. In many other cases, you have to simply put 2 things together without knowing how they are positioned in real space. But with DNA nanostructure template, you know exactly, and you can design exactly how they should be positioned with respect to each other. To give another example, I'll stop soon. To give you another example, people also used extended lattice structure to assemble carbon nanotube. This is related to the nanoelectronic application I mentioned a couple of days back. And what they did here is using the DNA break approach to create long channels on a 2-dimensional DNA base. And within the channel, there are DNA capture sequence. And at the same time, they functionalize carbon nanotube with short DNA strands or handles. And, and after that, the DNA hybridization would recognize and capture carbon nanotube into those channels. And by doing this, they can create very high density of DNA. And if you look, carbon nanotube array. So if you look at the SEM image here, here is the channel, and here you see with the carbon nanotube in it, you start to see this tubular structure embedded in the channel. I'll skip those and just mention the molecular positioning. Here, the tool is often based on the modification of DNA strands. When you make this DNA nanostructure, you can replace any one of those strands with chemical functionalized version. Okay. So what that means is when you make origami structure, for example, you have the possibility to add a chemistry functional group onto any one of the 300 strands. And you know exactly where those strands would be because you designed those sequence in a computer program. So what you can do is you can precisely position 2 functional groups at specific location on that 2D platform. And then at a later time, you can attach other things. There are a lot of organic chemistry you can use. You can attach other things onto that DNA sequence. And this R can be another molecule, can be another fluorophore, can be another protein, can even be another nanoparticle. I think I should probably stop here. I'll only mention drug delivery applications where you can use DNA nanostructure as a container. You can modify the surface with functional groups that recognize certain receptor, and that can create targeted interaction with cells. And once everything is into the cell, it can undergo digestion by enzyme in the cell, and that can release whatever drug you're intended to load. There are different formats of this. And another way of targeting drug delivery is you can create channels. You can create a long DNA tube, and you can anchor this tube onto the lipid bilayer. And in some cases, you can even add a lock, a blockade at the top of the channel. And you can also add a complementary DNA strand. And by having that complementary strand, you can release this blocking single strand so that the channel is open. Or if you don't have it, then it will be blocked. So I think, apologize, have to skip some of the slides, but hopefully you'll see more of the applications side of things in my next talk. Again, thank you for you taking your time to participate, and be happy to answer any additional questions.
Thank you, Professor Liu, for a very insightful and interesting lecture on DNA nanoparticles and how the scaffolds and strands of DNA can be used for designing some new materials for specific applications like drug delivery, tissue engineering, etc. Now the topic is open for discussion. If you have any questions from the audience, both offline and online, they can discuss. For online participants, they just write down their questions in the chat box or in the WhatsApp group, and somebody will moderate from the organizing team. And offline participants can ask questions directly. I have seen there are a few questions in the WhatsApp group as well. Aditi, can you please ask? Can you please read those questions?
So, how regular structures are made with DNA and how uniformity of shape is maintained?
How regular?
Regular structures are maintained with DNA and how uniformity of the shape is maintained.
So by regular, um, I presume maybe you're talking about periodic structure. I'm not sure exactly what regular means, but I can certainly comment on the second part. Okay, so let's see. I'll start sharing my screen. Okay, so first of all, on the stability, I have more experience with the stability of individual origami structure. I'm gonna show my pointer. Okay, so those individual origami structure, once they are made, they are stable in water, in the buffer, at room temperature, at least for a week, based on our experience. Now, if we freeze them, they can survive a lot longer. Now, there is some damage as a result of freezing, that because those are large structures, so the formation of ice crystal sometimes would damage them. Now, once they are deposited on a solid substrate, they maintain their shape for much longer time. In terms of stability, you can view them as the same as any other DNA double helical structure. They have a melting temperature. As long as you don't heat up your temperature above their melting temperature, they will be fine. They are also biological in nature, so if your environment has enzymes that chop up DNA, then that certainly would be bad. So in terms of stability, it would be the same as any other biological, any other DNA material in general. So the other part is
How uniformity of shape is maintained. And for structures, he's asking about regular shape like triangle, trigonal, bipyramidal, etc.
So there are a variety of ways to make them. If you look at this regular structure here is a rectangle, and all you need to do is to fold the DNA in this way. That's all you need to do. If you fold it this way, you'll get a rectangle. If you fold it a little bit longer, you'll get you can get square or rectangle. And the pattern here actually shows you how the folding happens. So the color, the shade of the color tells you which part the folding started first. So I think from the purple is one end and the yellow is the other end. So in this case, if you look at the square one, the folding actually goes just like this. If you look at this one, for example, it actually folded in two parts. So it's a rectangle structure, but you fold one side of the rectangle first, and then you go to the other side and fold the other side of the rectangle. So it goes like this, and then do it like this. Okay.
Sir, how we ensure that the functional groups are positioned on right place?
You do not have to, it's probably easier if I use this as an example. So this is a single-tiled approach of making DNA nanostructure, but the idea would be the same in that you have a lot of short DNA strands, but each short DNA strand knows where it needs to be in the final structure. Because if it's in another place, then the hydrogen bonding doesn't work out. So it has only one option, and you know exactly where it's going to be. So when you make the structure, you can chemically modify that strand and only that strand. And if you do that, you're going to install the chemical functional group only at the position that is supposed to be for that DNA sequence. Did that answer your question?
Sir, my first question is, sir, what parameters are involved which regulate their shapes? And sir, the other question is, sir, what about other biomolecules which are also available now? RNA biology is taking place. So, sir, have you tried RNA as a molecule in nanotechnology?
I think the first question is what parameters are involved in designing a shape?
Yes, sir.
Okay, so there are different approaches again. Now, if we look at the origami approach, then the consideration is when the fold happens. So that's the only thing you need to think about, when the fold happens. You can just think about a very long wire. You can fold the long wire 300 times in the XY plane. If you have the complete freedom to do that, what can I do to make a triangle shape? There are many different ways I can fold it, but I have to fold it in a way that it goes along the triangle shape overall. So I have to impose restriction that it has to fold in certain way. Now, the good news is there's a computer program to do that, taking into all the helical structure constraint of double-strand DNA. Now, in the case of single-stranded tile, the shape is even easier to handle because you can simply remove some of the DNA strands. The absence of that DNA strand in the final structure would create the shape.
Sir, so for this we use DNA whose whole sequence we know very well. So for that the complementarity of DNA is utilized for this folding.
So in the origami approach would be the complementarity, and we use the complementarity to fold the long strand. In this case, while still everything's underlying principle is DNA hydrogen bonding, but in this case you remove some of the strands. So example would be here, this triangle structure, and versus the whole rectangle structure. If you remove half of the strands in the collection that produce the rectangle, and if all the strands you remove are supposed to be here, then you create a triangle.
Sir, what about other macromolecule like RNA?
I think you asked about.
Sir, like what about RNA?
So the question is, what about RNA? And that's a very good question. So people have used RNA to create RNA nanostructure as well. Same kind of concept. The major difference being RNA is much more chemically liable. So chemical stability of DNA is much worse than that of DNA. So as a result, the research on the RNA nanostructure is not as widespread as that of DNA nanostructure.
Thank you, sir. Thank you, sir.
How I can, okay.
How did you use origami to make? How did you use origami to make DNA origami structure?
I'm not sure what. Oh, so you mean the actual process of making them?
Sir, one slide belongs to DNA origami structure. Then how you use origami, that is mathematics part. How you use origami to make this type structure?
How? I think you're asking me how do I use origami to make the origami structure?
Yeah.
I'm not sure what. Trying to understand the angle of the. So if you're asking about the actual process of making the structure.
DNA origami, one slide belongs to DNA origami. That is related to that.
Oh, something happened.
Yeah, that is.
This one?
Sir, that is. Previous. Yeah.
Previous one. This one?
Sir,
or this one?
sir previous. That is.
This one?
Yeah.
So your question is, how do I use origami to make the origami structure?
G. Yes, sir.
Well, I'm not sure what. Okay, I guess the origami would be the analogy the authors are making between folding paper versus folding DNA. The concept here is you fold a DNA long DNA sequence multiple times to create geometric shape. That is just like you fold paper into paper origami. So here you fold DNA into DNA origami. But I'm not quite. I don't think I get your question. If you can rephrase your question, maybe that'll help me.
Okay, sir. Okay. Thank you, sir.
Yes. So we have an online question also. How DNA is protected from WBCs in body for drug delivery before reaching the target?
Yeah. So the question is how the DNA nanostructure are protected from a lot of bad things in your human body before it's able to deliver drugs. That's an excellent question, and this is something. Let me see. Okay, this is something that people actually thought about and started looking into addressing them. The As you know, there are a lot of enzymes in your body. So a lot of enzymes are designed to degrade foreign objects and to attack foreign objects. So DNA nanostructure has the advantage of creating this ideal shape. You know, you can create a container, for example, to load the drug, to protect the drug, but also it has the problem because its DNA may trigger a lot of unwanted response, immune response in particular from body. And in addition, it's a biological material, so the degradation of biological material within human body is always a concern. Okay. What has been done, so people recognize this and they have thought about different ways of enhancing the stability of DNA structure. And this one, this is one of such examples where they attached lipid bilayer structure on they coat DNA cage with lipid bilayer structure as a way to mask the DNA material from being detected and attacked by enzymes and other proteins in the system. So it is a very a significant concern in my view, and in the future, whether we should use those native DNA as construction material for drug delivery, or we should use them as solely as a mechanism to do fundamental study. I see the possibility of both, but certainly the immune response and the stability needs to be addressed before this can be put in real use. Thank you for the excellent question.
Thank you for very nice lecture. Now we are proceeding for tea. We'll be back here again at 11 a.m. Today we have a short tea break.